Linkage first, right? So when we say linkage, this is actually the presence in the same chromosome of two non-allelic genes affecting two traits, okay? So linked genes violate Mendel's law of independent assortment. So how? So instead of segregating, okay? Instead of segregating and recombining in a random manner during gamete formation in your meiosis, the genes are inherited together. So as you can see here in the gamete, so the genes are inherited together as a single group, and this single group is called a linkage group. So this one is a linkage group, this one is a linkage group because dominant genes A and B are inherited together. Recessive genes A and B are inherited together. So there is no crossing over in here. And because of that, there is linkage. But in here, there is crossing over. So the chromatids, sister chromatids, there is crossing over in these homologous chromosomes. So we have here a random combination of genes as a result. Right? Now, I have mentioned a while back a linkage group, alright? So actually, a linkage group is simply the group of genes located in the same chromosome, okay? So this is the group of genes. This is one group of genes located in the same chromosome, and this is another group of genes located in the same chromosome rather, okay? Would like to correct myself. This is a group of genes located in the same chromosome, okay? If dominant gene A and recessive gene B is inherited in this one, then there is no linkage, okay? That is not linkage group because the dominant gene A and the recessive gene B are located on different chromosomes, okay? Ang tinatawag lang natin na linkage group, these are group of genes located in the same chromosome. So I hope I have made myself clear. Alright? So during prophase 1 kasi, the segments of the homologous chromosomes usually cross over, right? Or the exchange segments. So the farther the distance between genes in the chromosomes, the more likely that crossover will take place between them. So kapag mas malayo daw, kunwari itong gene A, mas malayo siya kaysa mas malayo ang distance niya sa B, then more likely that crossover will take place in them. So the closer the genes, kapag mas malapit ang genes, kunwari itong B and C, right? So malayo ang gene A sa gene C, kaya walang linkage na naganap. Now in here, the gene B and C, they are closer together. There is greater probability that they will be inherited together, right? Than those located farther apart. Of course, kung mas malayo ang genes sa isa't isa, edi ibig sabihin, mas mahirap na magkaroon ng linkage. Alright? Yun lang ang ibig sabihin. Now, how do we determine if there is linkage or if there are linkage groups present? So the method used to test linkage of genes is to backcross a double heterozygote animal to one that is homozygous recessive for both pairs of genes. So ito siya. You backcross a double heterozygous animal. So this is a double heterozygote animal. And this is a homorecessive for both pairs of genes. Okay? That is to test for linkage. Okay? So, if there is no linkage involved, ibig sabihin, the cross followed Mendelian genetics. The gametes will randomly segregate and independently assort, and as a result, 4 phenotypes will appear in the offspring in approximately equal numbers. So, if you remember your Punnett square, so ipapanit square mo lang itong cross na ito, ang magiging resulta is that equal lang ang equal lang ang distribution ng phenotypes ng offspring. Okay? Kapag walang linkage because it follows the laws of Mendel. Alright? So we have here a table showing, ito na yung sinasabi ko kanina, na kapag walang linkage, so this is the progenies. These are the progenies of the cross double hetero, and homorecessive for the two traits. So, say for example, this gene P codes for poledness and the gene B codes for coat color. So, yung dominant ay polled, dominant gene is polled, recessive gene is horned. The dominant gene B is for black and the recessive gene B is for red. So, if you cross these two individuals, kapag walang linkage, ito ang result. Okay? 4 equal phenotypes. So, ang unang phenotype, we have polled black, we have polled red, horned black, at saka horned red. Yun ay kapag walang linkage, kapag ang cross ay pinalo ang laws of Mendel or Mendelian genetics. So, ito, kasi makita tayo ito, with linkage, there is an equal distribution of phenotypes. Okay, so we will discuss this part a little later. So let's move on to the next slide. Okay, so with linkage na tayo. So actually, there are two types of linkage. Una, we have coupling linkage. Pangalawa, we have repulsion. Alright? So if you recall your Agri 21, sa coupling, nai-inherit together ang dalawang dominant gene. Right? Sa linkage kasi, we are talking now of two genes. Alright? Two pairs of genes, rather. So, in here, sa coupling linkage, dominant genes are inherited together. Ganon din sa recessive genes. Alright? Sa repulsion naman, yung opposite. Okay? Dominant and recessive genes are inherited together. And ibig sabihin nito, kapag dominant and recessive genes are inherited together, yung dominant and recessive nasa iisang chromosome. So, located sa isang chromosome. Alright? So, example, dominant P, recessive B, nandun sila sa iisang chromosome. Yung example natin kanina, magkasama yung dominant genes P and B. So, yung example natin kanina ay isang example ng coupling linkage dahil magkasama ang dominant genes P and B. Ngayon, sa repulsion linkage, iisa lang ang ibig sabihin nito. Dominant and recessive genes are located on the same chromosome and as a result, they are inherited together. Okay? Another, if there is no crossing over. Okay? So, with linkage, mayroon tayong dalawang types. Either walang crossing over or meron crossing over. So, unahin natin yung walang crossing over. So, ibig sabihin, complete yung linkage. Okay? So, that if there's no crossing over, then linkage is complete and close. Alright? So, we use again the same trait. So, poledness again and black and color of coat. Okay? So, dito, if there's no crossing over following coupling linkage, may inherit together yung dominant genes. Pati din yung recessive genes. So, say for example, meron tayong parental generation. Okay? Parental generation of two purebreds. So, two purebreds ito. Ang una, polled black. Okay? Ang pangalawa, horned red. So, we cross polled black and horned red. Ang F1, we have a double heterozygous animal with a phenotype polled black. Okay? Now, I mentioned earlier now that to test for the presence of linkage group, we backcross this double heterozygous animal to the homozygous recessive parent for both traits. So, yun na yung recessive gene P and then recessive gene B. Okay? So, we cross this. This produces now your F2 generation. So, if you remember your Punnett square, ganito, to get the gametes of this genotype, we just follow law of segregation or law of independent assortment. Okay? So, the gametes of this genotype are dominant P, dominant B, dominant P, recessive B, recessive P, dominant B, recessive P, recessive B. Yun yung gametes of this heterozygous animal. itong homozygous recessive, iisa lang ang gamit. Okay? So, the gamit is only recessive P, recessive B. So, I repeat, if linkage is complete and close, and if linkage is coupling, so coupling tayo dito ngayon, ang mai-inherit together ay yung dominant genes. And then, may inherit din together ang recessive genes. Okay? So, dominant genes go together, recessive genes go together. Alright? So, since dominant genes go together, this will be inherited together. Okay? This will not be inherited together. This will not be inherited together because this is a combination of dominant and recessive genes. So, these two. Okay? So, ito yung ma-inherit together. So, the dominant P, dominant B, recessive P, recessive B, inherited together. So, you combine these two, you produce a genotype, double hetero individual. Ganito din dito. So, you combine these two gametes, you now produce a homozygous recessive. Okay? So, ito na yung ratios of the genotypes. So, we have here 4:4 or 1:1. Okay? That is, if there is linkage, if the linkage is complete and close, following coupling linkage, polled and black cattle, as well as horned and red cattle are inherited together. Okay? So, polled and black and horned and red are inherited together. So, if you recall, polled and black, these are the dominant phenotypes. So, horned and red, these are recessive phenotypes. So, they are inherited together following coupling linkage. So, therefore, no horned black, no polled red. Okay? That is inherited. Okay? So, ito na yon. Ito na yung no crossing over. Okay? With linkage but no crossing over. And no crossing over. Okay? We will discuss the last part of the table in the next slide. Okay? So, with independent assortment, we have 1:1:1:1 ratio. That is the expected. Diba? Following Mendelian laws of inheritance. But with linkage, these are the results. Okay? Because, sabi natin kanina, we follow the coupling linkage, the dominant genes and the recessive genes are inherited together. So, dominant genes inherited together, recessive genes inherited together. So, these are just sample frequencies. Okay? Sample frequencies lang ito to illustrate how linkage happens. So, what does this mean? So, these are the parental combinations. So, the parental combinations of gametes will be more frequent. Diba yung parents kanina, if you remember, is a double heterozygous animal and a homozygous recessive animal. Okay? So, these are the parental combinations. These parental combinations of gametes will be more frequent than the recombinant. Ito yung recombinant. Tinawag silang recombinant because dito nangyayari yung recombination. This is where crossover took place. Alright? So, this is attributed, of course, to the crossing over during prophase 1 of meiosis. So, ano yung tinatawag nating crossing over? As a recall, crossing over is an exchange of parts of homologous chromosomes during meiosis prior to the formation of gametes. So, this allows for new combinations of genes or recombinants. So, di ba? From the previous slide, kanina wala. 0 ito. Because complete yung linkage. And walang crossing over. But because of crossing over for this second scenario, merong recombinant. Alright? So, as a conclusion, all 4 phenotypes appear in the next generation, but 2 appear more often than the other 2. Yun ay with crossing over. So, linkage with crossing over. Okay? Yung 2 appear more often, yun yung, again, I repeat, parental combinations. Yung appear less often, yun yung recombinant. So, ito na siya. We have 45 polled black, 5 polled red, 5 horned black, 45 horned red. Okay? So, ito yung parentals, ito yung parental combination. So, parentals because, bakit tinawag siyang parentals? Because the parents are or have the same genotypes with that of the parentals. So, pareho yung genotypes nila. Alright? So, how do we get for the percent crossing over. Since we talk about crossing over, paano natin makukuha yung percentage of crossing over? So, the number of recombinants between loci is used to calculate distances for each pair of loci. Okay? So, the percentage of crossing over among all offspring of a mating is also called map distance. So, yung percent crossing over, also called map distance. Alright? So, it is computed as the number of individuals per crossing over occurred divided by the total number of individuals or offspring. So, you multiply it by 100 to convert it to percent. Alright? So, each map unit is also called percent, also called distance, also called Morgans. So, in honor of Thomas Hunt Morgan. So, Thomas Hunt Morgan. So, sa example natin kanina, ilan yung crossover? So, the number of crossovers can be determined from the number of recombinants. Okay? So, meron tayong 5 plus 5 recombinants kanina, we have 10 crossovers. Divided by number of offspring, meron tayong 45 plus 5 plus 5 plus 45, that is equal to 100. And then, we multiply it by 100 to get the percentage. That will be equal to 10%. So, canceling 100, 100, you have 10% crossing over. So, there are 10 map units, so, or distance, or Morgans. So, 10% or 10 map units or distance or 10 Morgans, these are all accepted as answers. Okay? So, it is important to note that closely linked loci show fewer recombinants than loci far apart on the chromosome. So, sinabi ko kanina na kapag ang genes ay mas malapit sa isa't isa sa isang chromosome, there is lesser chance of crossing over. Yun ang ibig sabihin ng unang bullet. So, closely linked loci show fewer recombinants than loci far apart on the chromosome. So, ibig sabihin daw, kapag mas malayo ang chromosomes, kapag mas malayo ang genes sa isa't isa, sa same chromosome, ibig sabihin, there is a higher chance of crossover. But if mas malapit ang genes sa isa't isa, there is lesser chance of crossover. So, ibig sabihin, malaki ang chance na there is linkage. Okay? There is linkage of genes. So, if two loci are 50 or more map units or crossovers or Morgans apart on the chromosome, the probability of multiple crossovers is so great that the genes of the two loci are essentially unlinked and therefore segregate independently. So, yun din ang ibig sabihin. Okay? So, kapag daw mas malayo ang dalawang genes, okay, ibig sabihin, there is higher percentage na unlinked or mag-crossover, mag-exchange yung segments ng dalawang chromosome. So, that will follow Mendel's law of independent assortment. Okay? Or law of segregation and independent assortment. Therefore, the maximum observed crossover frequency is 50%. So, kapag nag-compute ka ng percent crossover or map distance, dapat ang maximum lang na makakompute ay 50%. Okay? If the computed value is higher than 50%, check your solution. Okay? Kasi dalawang genes yung kino-compute mo sa isang chromosome. Okay? Dalawang loci rather. Okay? That would be all for this video. I am your teacher Hazel. Thank you very much for listening, and I hope you learned something.